And now we're going to talk about K-means. So this is an unsupervised method for clustering. And that requires you specify the number of clusters in advance. So clustering actually is a broad set of techniques for finding subgroups of observations within a data set. When we cluster observations, we want the observation in the same group to be similar and observation in the different groups to be dissimilar. So because there isn't a response variable, this is an unsupervised method, which implies that it seeks to find the relationship between the observations without being trained by response variable. So actually clustering allows us to identify which observations are alike and potentially categorize them. So K-means clustering is the simplest and the most commonly used clustering method for splitting a data into a set of K groups. So for data preparation, the rows are observations and the columns are variables. So in our case, the rows is individuals and the columns are the genes. And any missing value in this data should be removed or estimated. So if there's NA, just use NA.omit, or you can just replace this NA with some number. And the data must be standardized to make variable comparable. So we want the standardization to, we want the standardized data to have a mean around 0 and standard deviation of 1. So here is a cartoon to showing K-means with 3 centers. So we can so firstly we pick 3 random data from the data set. And we calculate the mean for the data points assigned to that cluster. And then we took the mean as the center and redefine the clusters and take the mean again to reassign the center. So after a couple of the iterations, we're going to get to a local minimum for within cluster distance. So this will not provide like the best separation, whereas only going to provide a local optimal. So for some of these data, there might be like the method might trap into a local minimum, and that might not be a good choice to do K-means. And how should we I'm going to talk about how should we choose for the K in a later session. Here, just showing you how to see the distance between samples. So we can use this getDist function to get the samples. We first need to scale and omit the data, as I said, omit the NAs, as I said, and then we use the getDist to do to find the distance between each data points. And the default distance in R is Euclidean, so we're calculating the Euclidean distance. And we can see a visualization like this. So for some of them, maybe the red ones, these data points are further away from each other, and for some of them, they're closer to each other. And this basically shows us how so this ones that's closer to each other is going to be clustered together, while this ones that's further away might not be the same cluster. And there's another way, another visualization you can use, which you can just delineate this boundary here, so you can have a better visualization. Yeah, just an option for you guys for a better way to present your data. And as I said, you can have different centers for different data sets. Here I'm showing you not only the 2 clusters, I'm showing you 3, 4, and 5. So you can see they can do a pretty good separation. But like for here, maybe 5, if you have 5 clusters, these are not too separable, which is not ideal. Yeah, so yeah, you can see like whether the K is good from this plot, or you can do something like this, where you plot the iteration, the center numbers with the within cluster distance. So we can see here the distance, the within cluster distance decreases as you increase your number of centers you pick. So let's say if we have if we only have 10 data sets and we pick the k equal to 10, then we're going to reach to the within cluster distance to 0, because each of these data points is going to be a cluster itself. But there's no much information we can get from them because we are just, you know, we cannot see the relationship between data points. So we don't want to have a really large k, but we want to have we want to do as good separation as we can. So for example, in this graph, maybe 4 is the turning point, where you have a relatively big decrease of the within cluster distance, but you still preserve some of the relationship between data points. Yeah, you just try to do something like this and find the optimal k you can get. And now I want to talk about the hierarchical clustering, which is an alternative approach to K-means clustering. Except for hierarchical clustering doesn't need any pre-specified number of clusters, and it generates this tree-based representation of the observation called the dendrogram. So as we can see from here, this is the hierarchical clustering. So there are a lot of different methods you can pick for this each cluster. You can check online how these are different. This is going to be a whole lecture, so now I'm not going to go deep into it. Yeah, but we can just show you guys briefly what this look like. So for example, we have this dendrogram, and if we want to have like let's say 4 clusters, we can just cut it from here. So we know like these are in one cluster, and whereas these are in another cluster. You want to only have 2 clusters, we cut over here, and we can see yeah, there is well separated. So not only that, hierarchical clusters doesn't require a predefined cluster number. It can also show like within each of the clusters which 2 samples are closer together. Let's say we know like all these data points are clustered in the same category, but we know these 2 are more similar than this one from this one. So that dendrograms gives us more information that we need, and it's more flexible for us to define the clusters.